Okay. Let's see. All right. Just making sure my microphone was going. Let's talk about now. Let's get into how we go about actually identifying some of our prokaryotic cells, and we're going to talk about the domain bacteria here because we know more about them, and we'll actually be looking at some bacteria specimens in lab. So bacteria give very little information for identification without using some kind of molecular technique to sequence DNA. So this is can be a little tricky in the lab, and it historically has been tricky before we had genetic techniques at our disposal. But there are some ways to do it. And we are going to be kind of, first I'm going to talk about sort of how we go about, some ways to recognize different groups of bacteria, and then we're actually going to talk about some different groups of bacteria that we see on this tree. So we can do this a few different ways. We can look at the shape, go back here, the shape of an individual cell or the shape of a colony of cells. We can look at the way that these cells organize themselves, such as in perhaps chains or clusters. We can look at different cell surface features, and we can look at the motility of a cell, and these all give us some clues. So let's talk about shape. So this is one thing we'll look at in lab. So there are three, well, there are many shapes we can run into, but three really common shapes that we run into are what is called the cocci. The cocci, which is a spear-shaped bacterium. The bacillus, which is a sort of a kind of a rod-shaped bacteria, and the spirillum, which is a spiral-shaped bacteria. And we'll look at all of these different shapes in lab. So we also get some common organizational arrangements. So what we call the diplo, so not surprisingly, diplo is referring to two. So whenever you see two cells together, and this is a common way they'll organize, we have sort of the prefix diplo, and then we would also at the end signify their name. So if you have a diplococcus, what does that tell you? Well, it tells you I've got arrangement of two, and it's a spiral-shaped bacteria. We can also have what's called strepto, a strepto arrangement. A strepto arrangement is when you have a chain. So streptococcus would be a chain of spears. Streptobacillus would be a chain of rods. And then we also have what's called the staphylo. The staphylo arrangement, which is kind of like a cluster, or a packet, sometimes they call it. So we have our staphylo arrangement, which is clustered. So a staphylococcus would be a cluster of spheres, for example. So we have those three common arrangements. We also can sometimes learn something about our bacteria by looking at the colony morphology after culturing. So what do I mean by culturing? Well, if we have a agar plate, so this is a, so we take a plate and we put on that petri dish plate some kind of medium. We oftentimes use agar, which is derived from seaweed. It's just kind of a growth medium, and we basically put our bacteria on here and we allow it to grow into a colony. So these blotches you see are not individual bacteria. These are colonies of many, many, many bacteria. And the morphology that these colonies take can tell us something about the bacteria. So we can have many different forms of colony morphology after culturing, and this is another thing that we're going to be looking at in lab. We can describe our colony form in terms of its shape, whether it's circular or irregular, filamentous, its elevation, whether it's raised or convex, and also its margins, so which would be kind of the edge of the colony. So this is somebody's handprint. It's kind of gross, but it's kind of cool, and kind of demonstrating. So basically, they took this agar, had somebody stick their hand. They allowed the bacteria and divide through binary fission, and look to see what the resulting colony was. So that's another way we can learn about our bacteria. We can also learn bacteria by observing cell surface features. So this would be looking at an individual cell. So almost all have a cell. There are a few exceptions that we'll talk about. The cell wall is really important in maintaining cell shape and in protecting the cell from the outside world. The bacterial cell wall is very different from eukaryotic cell walls. Bacterial cell walls contain something called peptidoglycan, instead of the cellulose or chitin we might see in a eukaryotic cell wall of a plant or a fungi. Peptidoglycan is a polymer made of modified sugars that are cross-linked with polypeptides. So they do have a cell wall, but it's very different in structure than what we see in our eukaryotic cells. Now, another thing about prokaryotic cells and their cell walls is that they can be either what we call gram-positive or gram-negative. So what on earth does that mean? Well, there's something called gram stain. Gram stain, not gram strain, sorry, grams stain, which is a technique used to classify bacteria into two groups based on differences in their cell wall. So this stain will classify bacteria based on cell wall differences. Because the type of cell wall you have is going to affect the color you turned when we apply Gram's stain. So how does this work? Well, let's talk first about the two different cell walls you might have. So some bacteria that are known as gram-positive bacteria have a plasma membrane on the outside of their cell and then a very thick peptidoglycan layer. Other bacteria, that we would call gram-negative bacteria, have a very different arrangement. They have a plasma membrane, and then actually what they've got is a thin layer of peptidoglycan and then another bilipid layer sort of sandwiching that thin peptidoglycan. So we kind of have this double membrane situation with a thin layer of peptidoglycan in between. So a very different structure. This difference in structure has an impact on the way that it takes up particular dye treatment. So the way that this is going to work, first, now keep in mind, if you just have and you're looking at them under a microscope or something, they're just going to look transparent. You're not going to see anything. The thing we do for is we apply a purple dye called crystal iodine. So we're applying two different things. And then we rinse alcohol. This is where we're going to see some differences. So for the gram-positive with that thick layer of glycan, they hold on to color. The gram-negative do not. So that peptidoglycan help kind of hold in that thick glycan, helps hold in that purple color, and those other bacteria would hold it in as well. And we'll talk about this more. But anyway, what happens, so colorization step where we do an alcohol rinse, the gram-positive has stayed purple, the gram now clear. But we still need to be able to see the gram-negative. Apply what's called a counter stain that is red. And what happens is, purple gram-positive bacteria, you can't see them. The purple covers it up. But in gram-negative bacteria that were rare, it takes up this counter stain. So at the end of everything, the purples are our gram-positive bacteria, and the are our gram-negative bacteria. So this stain tells us about the differences in their cell wall. And so again, gram-positive, let me just write this down. So gram-positive, we have a simple cell wall with a large amount of the peptidoglycan, and gram-negative, we have I guess I shouldn't write a minus sign there, that's confusing. We have a more complex wall with a small amount of peptidoglycan that is sandwiched between two membranes. Another thing I wanted to mention, on the gram-negative cells, on this outer membrane of the gram-negative cells, we have, I don't have a good picture of it here, but on the outer membrane of the gram-negative cells, we have these things called liposaccharides hanging out, which is a combination of sugar and lipids on outer membrane. Which can change the characteristics of the cells. So these liposaccharides give them certain properties. A lot of times it can actually make them more virulent, so these gram-negatives can sometimes be quite nasty ones. Okay, so I'm going to end this video clip here and pick up on the next one, where we're actually going to talk a little bit about some of the different bacterial groups and learn a little bit of taxonomy. So get ready to hear a bunch of different names.